you. All right, so today is all chapter 11 from beginning to end. And it sounds like a lot, and as you read through, you will find it is a lot of detail that you will not be tested on. The purpose of today is to run through a number of diagrams, many of which look as confusing as anything we have talked about this year. But I just want to explain what the book is trying to illuminate you on. Everything we talk about today is related to your video project. It's all about cell communication, cell-to-cell signaling, and how messages and signals are sent throughout the body. So there's a number of things I'm going to highlight, but I'm not really going to go into much detail, especially when I don't need to. My suggestion to you is this, though. To have a good video project, you need to pull some stuff from here, from chapter 11, into your educational video. And regardless of the topic you chose, there are a number of different things you could pull in, and that's a major purpose that that's kind of your task for the video project, is connect back to chapter 11. Now, I will see you Monday, Wednesday, and Friday of next week. Monday and Wednesday, I want to, with those 2 days, get through all of chapter 12, which is the only chapter you'll actually be tested on. Just like today, on Monday and Wednesday, when I'm done talking, you can have time to work on your project. And then Friday of next week, our last day before Thanksgiving break, we should be in a good position to go down to the lab, get the microscopes out, and do a lab related to mitosis, which is a major theme in chapter 12. That means on Wednesday, we should be going through the last lecture of this whole semester. We'll be done with the notes. 3 classes from now, or 2 classes from now, not counting this one. And a lot of the time between Thanksgiving and Christmas will be dedicated to reviewing chapter 12, you making sure that the video project related to chapter 11 is done, and then we'll have a half test, and we'll turn in the project, and then the few classes we have after that, we'll look at viruses a little bit for your reading assignment, and we'll do some like practice AP exam stuff, and that's about it. So really this next 2 weeks or the rest of this week and next week will be pretty much all the new stuff, and then the rest of the semester will just be making sure you do well on the test and project. All right, so chapter 11, cell communication. The chapter starts out with this example of an impala, this deer-like animal, running from, I believe, a cheetah, which is the predator, one of the big predators for impala. And they are connecting this real-life, natural scenario to what is happening in your cells. All right? So when you or any other animal that has adrenal glands sees danger, true, clear, present danger, there will be a signal that your brain sends to your adrenal glands that sit on top of your kidneys. And your adrenal glands will begin to dump epinephrine into your bloodstream. Another name for epinephrine is adrenaline. Have you ever heard of adrenaline? Yeah. Yes, you probably have heard of a term like an adrenaline rush, whether it's from getting on a roller coaster or coming across a machete-wielding psycho in the woods. So, this is basically all of chapter 11, summed up in one simple diagram. Epinephrine is a hormone. It's also called adrenaline, so we can use either term. Epinephrine is a hormone, but more importantly, it's a signal that your body sends out in the form of a molecular signal. And cells throughout your body have proteins in the what? What's this? This is the membrane. It's a phospholipid bilayer. So there are proteins in, some of the proteins in your cell membrane will be receptor proteins. They are going to have a structure that can bind to a signal, in this case adrenaline, and when that signal binds to a receptor on the surface of your cell, it will cause a series of chemical reactions to occur, and at the end of that series, there will be some kind of change that has taken place in the cell that we refer to as a response. So essentially, chapter 11 just slogs you through a discussion of how signals can be received by cells, which we call signal reception. They can cause a series of chemical reactions to occur in response to that signal, which we call signal transduction. And then at the end, a change takes place in the cell, which we call a cellular response. So basically it's reception, transduction, and response. That's what chapter 11 is about. Now there's a lot of different receptors, and there's a lot of different ways the signal the receptor receives can be transduced. And then there are a lot of different possible responses that the cell can have as a result. And chapter 11 makes you painfully aware of all the different ways this could happen. But in every example, it's these three major steps that take place when a cell talks to other cells. One of the first times this was discovered was in yeast cells, when yeast cells reproduce or mate. So I want to talk to you about this a little bit. Your book goes into it, but it's a very simple and straightforward example of how cells can interact, create a response that serves a purpose. So you have two types of yeast cells, and I don't know why this is the naming scheme for these two types of yeast cells, but some yeast cells are known as type A, and others are known as type alpha. This is the Greek lowercase a, where we get our lowercase a from. So you have A yeast cells and alpha yeast cells. These are essentially like two genders of yeast. You wouldn't have male and female, you have A and alpha. Now, what scientists first noticed about these yeast cells, when yeast cells make more yeast cells, when they reproduce, is that A yeast cells, type A yeast cells, produce a chemical that they release when they're ready to reproduce. That's called A factor, because it's from the A cells. The alpha cells produce alpha factor, a chemical they make and release, and that alpha factor comes from alpha cells. And what they noticed was the A factor chemical would bond to receptors on the other kind of yeast cell, And the same happened for the alpha factor that the alpha cells release. So this type of yeast cell would create a chemical that the other type of cell could detect, and vice versa. And what they noticed was wherever you had a receptor receive a signal, the cell in this region would start to bulge out. It would create this kind of bulging to its surface. So the cell would kind of grow towards its mate. Eventually the two would grow into each other and combine into one cell that was a combination of both mating types. So they would merge as one. Now, what would happen from there is a process called meiosis, where this cell would then divide into 4 cells. That is the topic we start the second semester with, is meiosis. So I don't expect you to understand what that means, but essentially this process would happen, and eventually these 2 cells would merge into one, and it would divide into 4, and you have reproduction. You now have more yeast cells. But there was a signaling chemical secreted by one cell, read by another cell, and then a response that took place, which had a purpose towards reproduction. And I think that was in your reading for today. Now, there are some terms that we've already discussed, like in this slide. There are a few of them. So, again, like with yeast reproduction, a lot of things that happen in the living world rely on communication between cells. Now, sometimes cells are connected to one another, and you have these junctions where chemicals can just freely pass between these 2 cells. You actually will have little channels that kind of breach the cell membrane of 2 cells, and cells can just directly have chemicals exchanged between them. So they kind of communicate in that way. Do you remember this term, plasmodesmata? I briefly mentioned it in Unit 2, I think it was. And essentially in plant cells, you have breaks in the cell wall where plant cells can send chemicals to one another. Just these little channels that are kind of openings between 2 cells. They have this kind of ridiculously complex name of plasmodesmata, but it just means that there are kind of breaks in the cell wall where 2 cells that are connected to one another are able to share cytoplasm and chemicals in the cytoplasm. What we didn't talk about, but you saw in that Inner Life of the Cell video that y'all did your last little project on, is these cell surface molecules where you will have a protein on one cell that can actually reach out and make contact, direct physical contact with a protein on another cell. And when that happens, it elicits some sort of response. All right, do y'all remember that? They were like, in the video, they had these little protein arms that would like reach out, and then there'd be another protein that they would connect with. When that happens between 2 cells, you can have some sort of cellular communication. So we've kind of at least briefly seen examples of these or talked about these possibilities in the past. Your book, though, goes into a few other terms and a few other ways that cells can signal to other cells. And this is a diagram, again, from your reading, so you'll be able to see these terms. But you have what we call paracrine, synaptic, and endocrine signaling. And they're not really as complex as the names may make them sound. They're just ways in which cells can reach out chemically to other cells. So sometimes, in the case of, say, paracrine signaling, you have a cell that just releases chemicals in all different directions, and they're intended to stimulate a response in cells that are very close by. It's what we would call a local signaling. Para literally means kind of around, so when you say paracrine signaling, you're talking about a cell that is sending out chemicals to just other cells around it, very nearby. Your nervous system, nerve cells, all the cells of your brain and the rest of your nervous system, your nervous system uses this synaptic signaling very often, and that's when one cell is positioned and releases chemicals in a way that it targets another cell that's nearby. So you have one cell that you're targeting. We call that synaptic signaling, where essentially chemicals are released and they just drift across a gap to another cell that is the target of that signal. And then you have the example we started with in the first slide, where you have something like adrenaline, and that's what we call an endocrine signal or endocrine signaling. That's where hormones, you've heard the term hormones, we've talked about them a little bit. Anytime you hear the term hormone, this is going to be where a cell is going to release a signal into the bloodstream. And once that chemical signal gets in the bloodstream, it can travel throughout the entire body. And any cell, anywhere in the body, that has a receptor for that signal can have that signal sent to it and respond accordingly. And that's what happens with adrenaline. When you have an adrenaline rush, basically adrenaline is released into your bloodstream. It can travel to muscles throughout the body, to the heart, to cells in the eyes, and on the surface of your skin. There's all sorts of cells all over the body that are affected by receiving that signal. Now, regardless of how local or systemic the signaling is, and you're going to get kind of sick and tired of me talking about these three general steps, but this is the entire chapter. Regardless of how far away you have to send that signal, you still always have a molecule that serves as the signal, a receptor that receives that signal by binding to the signaling molecule. You will have some sort of chemical chain reaction known as transduction. And in the end of that chain reaction, something will happen in the cell that we refer to as the response. And we're about to go through some really dense examples of this general process. And I just want to show you these examples so that when you do your reading, you get a feel for what they're talking about and you're not just completely lost in all these different bits of terminology. So keep in mind, every slide I show you from here on out, somewhere there's a signal, a receptor, a chain reaction, and then a final response to the signal. Okay? So, keep in mind, there's only three major things happening here. Your book spends a lot of time, or a little bit of time at least, talking about what we call G protein coupled receptors. This is a special kind of receptor that shows up in a lot of different cells. Now, again, I'm not going to have a test question. I'm not going to have a test on this chapter, so there's not going to be any specific examples of this show up on a test. And at worst, on an AP biology exam question at the end of this year, they might reference this, but it's not like you're going to have to draw all this out and memorize it or anything like that. Okay? But some of you will pick a topic for the video project, and the topic you pick might involve one of these G protein coupled receptors. And you'll want to talk about them in the video if that happens. So, there is a special type of receptor that when it receives a signal, it is going to activate what is called a G protein. Normally that protein is inactive, but when the signal is reached, your receptor activates, and it's going to activate this G protein. This G protein actually moves along the inner part or inner side of the membrane and kind of bounces back and forth between two proteins. And when it hits an activated receptor, it is activated with a chemical called GTP. Now, let's play a little fun game here. What do you think the TP stands for? Triphosphate. It's almost the exact same thing as ATP, but instead of adenine, you have guanine. So it's not adenosine triphosphate, it's guanosine triphosphate. So a chemical that's very similar to ATP. But essentially, using this ATP-like chemical, this protein becomes activated. And when the activated G protein hits an enzyme, the enzyme becomes activated. And what do enzymes do? They speed up chemical reactions. So now that this enzyme has been activated, it will cause a chemical reaction to occur. Now look, there's a million different chemical reactions it could cause to occur at a faster rate. But there's some sort of chemical reaction that is not occurring, and then it will occur in response to this enzyme being activated. So basically you have a signal being received, some kind of transduction where you have this protein that is going to serve as kind of a middleman, and then the enzyme is activated and a cellular response takes place. Once that chemical reaction takes place, usually, like the phosphate group will be removed and things will go back to the inactive state and the response stops. If you pick a drug for your video project and the receptor it affects is a G protein coupled receptor, you'll need to talk about it. But there's other types of receptors. This receptor is known as a receptor tyrosine kinase. Wow, what a simple name. But it's still going to be the same basic concept. In the membrane, you have a receptor. Now, this is a weird receptor because there's actually two of them that are separated from one another until they both receive a signal, and then once they receive a signal, they actually merge into one unit, which they call here a dimer. Di meaning two, because this one single structure is actually made up of these two individual structures that come together. Once this dimer is created, ATP comes in and adds a phosphate group to these 6 different points on this combined activated receptor. Those phosphate groups that we get from ATP are going to activate proteins that cause some sort of chemical reaction to occur that wasn't happening before, and that is going to be the response, the cellular response. Eventually, they will become inactive and the whole response will shut down. But it's the same idea we keep talking about with this chapter. Signaling some type of change known as a transduction, and then eventually those changes, that transduction, results in a chemical reaction occurring that was not occurring before, and the cell will be different and will be responding as a result to the signal. If you pick a topic, if you pick a drug for your video project, and the receptor that that drug interacts with is a receptor tyrosine kinase, talk about it in the video. There is another type of receptor that you can find in cells. This is called an ion channel receptor. This is a little simpler. Basically, in this case, the receptor is a transport protein that can open and close. And all the signal does is cause that receptor to open. And when it opens, you can have what kind of diffusion? What's the term? Facilitated diffusion can take place. Usually in this case of an ion, like a charged calcium, a charged potassium, or a charged sodium ion. And when these charged ions move in or out, who knows, just from high to low, so they will cause a change to take place inside the cell, which is the response. Again, signal, transduction in this case would just be the opening of the channel and the flow of the ions, and then some response would take place. When the signal is gone, the channel closes and it stops. There is another example. By the way, with these ion channel receptors, if you pick a drug that affects or interacts with a receptor like this, talk about this in the video. Not all receptors are on the surface of the cell. Some receptors are actually in the cell, and the signal must pass through the membrane to get to the receptor. So this is a little different. This is how a lot of hormones actually work. For a number of hormones, you don't actually have the receptor on the surface. The receptor is inside the cell. In this example, when the signal activates the receptor, this activated receptor can actually move into the nucleus, stimulate the DNA to make a messenger RNA molecule. That messenger RNA can go to what protein-producing organelles? Ribosomes. And then a protein is made. That protein production is the response. Again, signaling, some sort of middleman event like the movement of this activated receptor to the DNA and RNA being produced. This is transduction. And then at the end, you have some sort of response. One of the topics you pick, or of the topics you could pick from, one of them, maybe the one you chose, interacts with a receptor that is actually inside the cell and causes some sort of mRNA production. That would be kind of unusual, I think, for the ones on the list that you had for that project. But if that comes up for your topic, make sure you talk about it in the video. Okay, so then the chapter chapter 11 moves into specifically talking about these transduction pathways, the chain reaction that happens in the middle between a signal being received and a response being produced. So you can see from this diagram that these transduction reactions, this chain reaction that acts like the middleman between signaling and responding, can be pretty complex. What you see here is a signaling that initiates this kind of set of reactions in which an inactive protein becomes active, which causes another different inactive protein to become active, which causes another different inactive protein to become active, and then that activated protein is what actually drives some sort of chemical reaction that serves as the response. Notice what is involved in this process that we see a lot of in biology. ATP. Yeah, a lot of the activation of an enzyme is actually going to be from ATP giving that enzyme a phosphate group. So your book talks about phosphorylation cascades, which is a fancy way of just saying ATP starts activating a series of enzymes by giving them a phosphate group. There are also enzymes that then rip those phosphate groups off and make that activated enzyme inactive again so that the signal that's causing the response can shut down at some point. You don't want necessarily to have this response keep going, so there's always kind of a feedback where you can inactivate these proteins and stop this specific event from happening. Your book goes into a lot of this information in the later part of the chapter. This is the exact same concept, except a slightly different pathway. Here we have this G protein again that gets activated. The activated G protein activates an enzyme. But in this case, the transduction is even more complex. This enzyme, in some cases, actually chemically alters ATP and converts it into a different chemical called cyclic AMP. What do you think the MP stands for? Monophosphate. That's right. So this 3-phosphate ATP is changed into a different chemical that only has one phosphate group. It serves as a continuation of this transduction event. This chemical will activate an enzyme, and then that enzyme will create a cellular response. I'm saying the same thing over and over and over again, aren't I? And that's what chapter 11 does. It's so repetitive. Ian Hankinson has to put his head down just to wrap his mind around it.
Don't worry, I was still listening.
Just for a moment, and he was still listening.
ATP giving the phosphate.
Oh yeah, that was the last slide. But still, it's signal molecule activating a receptor, and then a series of reactions that involve enzymes becoming activated, maybe causing some chemical changes that lead to other enzymes being activated before you get to some sort of final change that we consider to be the cell's response to the signal. Here's another one. It's really fun. Look at all this crap. Look at it! Now look, almost everything you see here is transduction. This is the signaling phase, which we have seen like 5 times already through these different slides. It activates a protein. That protein activates another protein. This protein activates an enzyme. This enzyme creates, through a chemical reaction, these molecules that we give these different acronyms to, and your book discusses and gives you the full name. You can look at them. But essentially at this point, once you create this chemical through this chain reaction, that chemical will then bond to an ion gate and open it. And then calcium that's inside the endoplasmic reticulum flows out and causes other proteins to become activated, and then you finally have some sort of cellular response as a result. All of that is transduction. And I know, look, you should feel overwhelmed by this, but there's something good about that. I don't want you to think that when I say that 2 cells are communicating, and I've been guilty of this, I just say like, oh, look, in this Inner Life of the Cell video, this protein reaches out and touches another protein on another cell, and they communicate. That's almost too simple. Now, I don't know that your book really had to go into all of this, but sometimes when you say that like one cell signals to another cell, it's not just cell got the signal, cell read the signal, cell changed. You have these various steps in between that have to happen before you get a final response. There are 3 slides left. We're going to finish them pretty quickly. The last 2 are actually, I think, much more interesting than all of this. Now, you might be asking, why so complicated? Why does the cell, when it receives a signal, activate something that activates something that causes a chemical reaction, that creates a molecule, that activates an enzyme, that then causes a reaction that is considered the response? Why all that middleman stuff? It seems inefficient. Well, this is a detailed look at the very beginning of this chapter where they talk about the impala seeing a cheetah and releasing adrenaline. And here's what happens. One molecule of epinephrine, one adrenaline molecule, out of all the adrenaline molecules you release when you see something that scares you or excites you, one molecule will activate one receptor. That's the signaling part. But the beauty of this transduction is that in the case of adrenaline, that one receptor will cause a few molecules to be activated, which causes, each one causes a few molecules of this cyclic AMP to be made. And then each one of these molecules activates a few different proteins. And each one of these activate proteins activate a few different other proteins. And what you're doing is you're amplifying the signal. So that when you get to the end, you have millions of activated enzymes that can be used by the cell to chop up polysaccharides into millions of glucose molecules. And what are these glucose molecules used for? What do you use glucose for? Energy.
Yeah.
Okay? So, you're walking through the woods in Canada, and a grizzly bear comes up upon you. Your cells are going to amplify this signal so that you can get a lot of glucose ready, because why are you going to need energy? To run or fight, but you're going to lose that fight.
You're not supposed to run,
I don't know what you should do with the grizzly bear.
I think you're done.
No, you're just,
I guess at the very least, if there's nothing to be done, you'll just be a sweeter meal with all that glucose you just freed up.
that would be pretty nice.
Yeah. So at least you can be like, oh, well, at least I taste better now.
Yeah, be nice.
But essentially, a lot of this chapter goes into this complex series of reactions that are meant to involve more and more and more and more and more molecules, so that at the end, one molecule of adrenaline is freeing up millions of molecules. That's actually like hundreds of millions of molecules of glucose. So you're really amplifying the signal. Now, I know we've been talking for a while, but I want to get through these last two slides because they give you an example of how cell signaling is used in a process called apoptosis. And I want you to understand what apoptosis is. A simple definition for apoptosis is that it's basically when a cell intentionally destroys itself. Your cells can basically be told by your body to end their existence, to break themselves down, and to basically destroy themselves. There are receptors, they call it death signaling receptors, where when activated, the receptor will inhibit an enzyme that suppresses other enzymes. And when it's inactive, these enzymes activate, and these enzymes are known as nucleases and proteases. Nucleases are enzymes that chop up DNA. Proteases are enzymes that chop up proteins. And so there are times in living things where signals are sent to certain cells, and those cells basically are told to digest themselves. Now you might ask, why would a cell tell another cell that it's no longer going to function and that it needs to digest itself? Well, and this is where the last slide comes in. You did this your age plus a few months ago in, I don't know if it's embryonic or fetal stages, but while you're developing in the uterus, your hands and feet at one point are just like solid flaps of tissue, okay? And apoptosis signals are sent to the cells along seams in that flap of tissue. And essentially what happens is you get these seams of cells that are told, destroy yourself. And what does that result in?
Fingers.
Fingers and toes.
Let's go.
Yeah. So your hands are kind of fingerless at one point, and then by destroying cells in, kind of lines or seams between where your fingers should be, your fingers actually separate out and your toes separate out into digits. This doesn't always happen properly. I once taught with a guy who apparently, while he was developing in the uterus, the seam between his pinky and ring finger, those cells never died. He had a thumb, 2 separate fingers, and then the 4th and 5th digits here were still fused together. And that is just a, I hate to use a, pejorative term, but it was a failure of some sort of signaling molecules not being sent to those cells to separate. So that's the use of apoptosis. The tail of a frog, as it turns from a tadpole into an adult, also undergo this process of apoptosis. And again, they added that in at the end of this chapter because this is done through signaling. That is chapter 11. That is all I want to talk about it, and I talked about it a lot. You still have to read it, but find where you can connect and implement some of this information into your video. Okay? I'm going to stop the video now.